Okay, let's unpack this. We're diving into the world of neuro-AI today. Where understanding our brains meets building smarter AI. There's this huge ambition, this dream of decoding biological intelligence, using it for better algorithms, maybe even things like prosthetic memory. You hear the buzz everywhere.
Absolutely. It's a really exciting space. But that dream runs headfirst into reality pretty quickly when you look at actual biological neurons. They're not the neat little diagrams you see in textbooks, right? Real neurons are, well, they're messy. They're variable.
Let's see how.
They just act differently moment to moment. They might spike in bursts. Sometimes they just go quiet when you wouldn't expect it. Definitely not simple, predictable circuits.
Right. So how have people traditionally tried to model these wild neurons?
Well, the classical approach involves really detailed simulations, solving complex equations, PDEs, partial differential equations. You build these intricate models, add parameters, run optimizations, and it can chew up, literally 600,000 CPU core hours.
Wait, 600,000 hours for just one neuron model?
Exactly. Just to match its specific behavior profile. It's incredibly slow.
Wow. That sounds like a massive bottleneck if you want to study, lots of them.
It is. It makes large-scale exploration really, really difficult. And these models, they're often deterministic. So to capture the natural variety you see in a real brain population, which is vital for function, you end up needing this huge collection of slightly different models, like a Hall of Famer.
A separate model for every little variation.
Kind of, yeah. And that approach just doesn't scale if you're thinking about simulating bigger brain structures like cortical columns or anything like that.
Okay, so the super detailed simulation is too slow, too rigid. What about using standard deep learning? The AI that's done amazing things elsewhere.
Yeah, people have tried that, but standard deep networks kind of hit their own wall here. See, neuron behavior, the voltage changing over time, it's about dynamics, complex relationships across time. Mathematically, it's like operating in infinite dimensional function spaces.
Okay, infinite dimensions sounds complicated.
It just means you're mapping whole functions, not just single points. Standard networks are great at learning fixed input to fixed output mappings point to point, but they struggle with that full dynamic function-to-function thing.
Ah, so they learn the voltage now based on the input now, but not the underlying continuous rule that generates the whole voltage trace.
Exactly. And they often don't generalize well.
Yeah.
If you change the simulation time step or give it an input current it hasn't quite seen before, performance can drop off significantly.
Right. So we've got these complex, messy biological targets, and the main tools are either way too slow or not really built for these dynamic function problems. Feels like we need a different angle.
We do. And that's where this other family of machine learning models comes in called neural operators.
Neural operators. Okay,
Yeah.
what's different about them?
Well, unlike standard networks learning point to point, an operator learns a map from an entire function to another entire function. So you feed it the whole input current waveform that's a function of time, and it gives you back the entire voltage trace waveform, another function of time.
A function in, a function out. Okay, why is that such a big deal for biology or physics?
Because these complex systems, fluid dynamics, weather, neuron firing, they are functions. They evolve continuously over time or space. Learning that function-to-function mapping directly lets these operators grasp the underlying physics or biology much better. And, crucially, they can slash simulation times dramatically.
So instead of just predicting outcomes for specific inputs, they learn the system's transformation rules across the whole input function space.
Precisely. They learn the mathematical operator describing the system. And there's actually a whole toolkit of these. There's the Fourier Neural Operator, which is really good for smooth global dynamics, the voltage traces, because it works in the frequency domain.
Like breaking down signals into sine waves.
Exactly. Then there are graph neural operators for handling irregular shapes, maybe like the branches of a neuron's dendrites. And even physics-informed neural operators, where you can bake known equations like the Hodgkin-Huxley equations for ion channels right into the learning process. Useful if your data is sparse.
Okay, so it's a specialized toolbox designed for these complex, dynamic, function-based systems. A step beyond classic deep learning for this kind of problem.
Yeah, that's a good way to put it. They offer capabilities that standard nets often struggle with when you're dealing with this kind of messy functional data from neuroscience.
Which brings us nicely to the focus of this deep dive, this framework called NoBLE, that stands for Neural Operator, with biologically informed latent embeddings. What's the core idea? How does it use these operators?
So the core idea behind NoBLE is actually quite elegant, even if the implementation is sophisticated. It's about packing relevant, high-level biological insight about the neuron's behavior into the front end of the network. You give the operator a compact numerical code representing that biological behavior, and that code helps guide the operator in predicting the full dynamics.
Okay, give me an example. How would that work for a specific type of neuron?
Sure. Let's take a parvalbumin interneuron, a type of inhibitory cell. They have pretty distinct firing patterns.
Hmm?
If you inject current, you can measure how its firing rate changes. Researchers found you can often boil down key aspects of this firing curve, its excitability, to just two numbers.
Two numbers. What are they?
First, the minimum current needed to make it start spiking reliably. That's its threshold current. Let's call it sigma 3R ether. Second, how quickly its firing rate increases right after that threshold of the local slope, or epsilon flow. These capture key features of its excitability and gain.
Okay, threshold and slope. So Nobel takes these high-level biological features and uses them as input, along with the actual current being injected.
Exactly. It takes those two numbers, ether to arth, encodes them using a special sinusoidal method, kind of like NERF if you know that from computer graphics. This encoding works well because the Fourier operator likes sines and cosines, so it works in the frequency domain.
Makes
Then this encoded biological insight gets combined with the encoded input current waveform, and the whole package goes through a 12-layer 1D Fourier Neural Operator, FNO.
So the FNO learns the input current to output voltage trace function, but it's guided or conditioned by these interpretable biological features, QQ and ether.
Precisely. It's not learning in a vacuum. It's learning a biologically constrained mapping. And there's some smart technical choices too. Like they carefully downsample the raw simulation data, the voltage traces from the PDE solvers. They go from like 25,000 time steps down to about 8,500.
Does that lose information?
They do it carefully with filtering, so it keeps the important stuff like spike shapes and timing, but makes it much more manageable for the network.
Okay. And you mentioned smart training data sampling.
Yeah, they made sure the model saw lots of examples of the neuron firing right around its threshold. They used a skewed sampling distribution for the input currents. This forces the model to learn the really important dynamics, the action potential fireworks, not just the quiet or fully saturated states.
Okay, so you feed Noble the input current plus these key biological numbers, oof, and the FNO spits out the whole voltage trace. What's the really big payoff here? You mentioned something about synthetic neurons.
Yes, this is where it gets super interesting for researchers, because those biological features, UTH and UTHR, form a continuous input space, what they call a latent embedding. You're not stuck with just the, say, 50 specific neuron models you trained on. The model learns a smooth map from this continuous feature space to the space of possible neuron behaviors. Meaning you can pick any point in that UTHR space. Even points representing neuron behaviors the model never explicitly saw during training. And the operator can synthesize a completely new, but still biophysically plausible, neuron's dynamic response on the fly.
Whoa, so you've learned a continuous landscape of possible neuron behaviors. You can interpolate between known types or even create slightly new variations just by choosing points in this feature space.
Exactly. You're generating synthetic neurons whose behavior is consistent with the biology the operator learned, defined by interpretable features, and you can do it incredibly fast.
Okay, let's talk speed. You mentioned 600,000 CPU hours for the old way per cell. How fast is Noble once trained?
The training itself is pretty reasonable, done on a single GPU, like an RTX 4090, maybe 350 APOX. The model small, 1.8 million parameters. But the simulation speed, that's the game changer. It takes those hundreds of thousands of CPU core hours and crushes them down into fast GPU batches. Simulate just one neuron, you get maybe a 230x speed up over the traditional solver.
230 times faster is already huge.
But if you simulate a batch, say, 1,000 different synthetic neurons at once, which you would often do for population studies, the speed up jumps to a mind-blowing 4,200x.
4,200 times faster. What does that actually mean for a neuroscientist?
It means research that used to need a supercomputer cluster and weeks or months of waiting is now doable on a grad student's workstation, interactively. You can explore variability, spin up synthetic populations, test hypotheses. It just opens up exploration in a way that wasn't possible before.
And the interpretability. You said the features NUTH and UTHR are meaningful. How does that play out?
Right, that's key.
Oh.
Because the latent space is built on these features, you can actually see what happens when you change them. Slide along the UTHR axis in the latent space and watch the synthetic neuron become more or less excitable. Slide along the UTHR axis and see its gain control change. It turns modeling from this kind of blind parameter hunt into an interactive design process based on behavior.
That's really powerful, turning the model from a black box into something intuitive. But how accurate is it? Does all this speed and interpretability mean you sacrifice prediction quality?
Surprisingly, no. When tested on the neurons, it was trained on using the PDE solver data as the ground truth. The predicted traces are incredibly close, like 2% error, using a standard L2 norm measure. They talk about microphone-grade fidelity for spike timing. The spikes happen exactly when they should.
Okay, but that's on the training data. What a generalization. Neurons it hasn't seen. That's the real test for synthesis, right?
Absolutely critical. So they held back 10 neurons from the training set. These neurons were still within the range of ether where the model learned, but it never saw their specific traces. When they generated synthetic traces by sampling the latent space near the features of these unseen neurons, the ensemble of synthetic traces statistically matched the ground truth behavior of those held out neurons really well.
So it can genuinely synthesize new plausible neuron behaviors. How does that compare to just, say, averaging the parameters of two different traditional PDE models?
Oh, that's a crucial difference. If you just try to interpolate the low-level parameters like ion channel conductances between two detailed PDE models, you almost always get garbage. Spiking thresholds go haywire, waveforms look totally unrealistic, the model might just break. Those parameters don't mix linearly to create valid behavior.
Right.
Nobel works because it learned the smooth mapping from the behavioral features to the entire dynamic behavior, the voltage trace function. It learned an interpolable space of valid dynamics, not just parameter values.
It learned the rules of valid behavior, not just averaging the ingredients. And you can use this to create whole populations.
Exactly. Once the model's trained for a neuron class, you can just feed it a stimulus and sample, say, 1,000 random points from the valid latent space. You get a cloud of synthetic voltage traces whose statistics spike with amplitude, firing rates, all that look almost identical to what you'd measure from a real population in an experiment.
It's a synthetic population generator.
Basically, yeah. Need 1,000,000 realistic interneurons for a big simulation. Nobly can generate their dynamic responses parameterized by their individual biological features in seconds.
That really feels like a paradigm shift. From slow individual modeling to fast, population-level synthesis and exploration. What are the real-world implications beyond basic research?
Oh, the ripple effects are pretty significant. Think about neuromorphic hardware brain-inspired chips. These compact, efficient neural operator models are ideal for putting realistic neuron dynamics onto those chips without the huge computational cost of PDEs. A stripped-down version could fit right in on chip memory.
So, faster, more realistic brain-like computing hardware. What else?
Brain-machine interfaces, or BMIs. They often need really fast models to predict neural activity in real time for feedback loops. That 4,200x speedup. That could be the difference between an offline analysis and a truly responsive real-time BMI system.
Real-time prediction for controlling prosthetics or communication devices. That's huge. And drug discovery. How does that fit in?
Right, so many neurological conditions involve changes in how neurons fire due to genetic mutations affecting ion channels. With Noble, you could potentially tweak the embedding parameters, the ether, to mimic the effect of a disease-causing mutation.
Create a virtual diseased neuron.
Exactly. Then pharmacologists could test virtual drugs on these virtual neurons in silico, screening thousands of compounds to see if any restore a healthier firing pattern. It could massively speed up early-stage drug discovery.
Wow, faster screening by simulating diseases and treatments. This really ties back to those big NeuroAI goals, doesn't it? Building models that bridge scales from ion channels to cognition, but doing it fast.
It really shows how these operator learning techniques aren't just abstract math. They directly tackle core challenges in neuroscience. And Nobly is one example, but other groups are pushing this too, using graph operators for dendrites, physics-informed operators for plasticity. The field is moving quickly here.
Of course, no breakthrough is perfect. What are the current limitations or challenges for Noble?
Good point. Right now, it's modeling one cell at a time, scaling it up to networks with interacting synapses. That's going to need more complex embeddings. Just two features probably won't cut it when you add synaptic dynamics.
Okay, handling synaptic interactions in networks is a next step.
Also, it's currently trained on data from those slow PDE solvers. It's mimicking the solver. The ideal goal is to train these operators directly on raw experimental data patch clamp recordings from real neurons.
Learn directly from the biology, noise and all.
Exactly. Cut out the middleman of the PDE model. And then there's interpretability. The two features, UTH and UTHR, are beautifully interpretable for that interneuron. But what about a more complex cell, like a cortical pyramidal neuron? It might need 10 or more features.
And interpreting a 10-dimensional latent space is much harder than a 2D one.
Much harder. So maintaining that clear interpretability as complexity increases is definitely a challenge.
But these sound more like engineering problems to solve, not fundamental roadblocks.
That's how it feels, yeah. More like the next set of research questions and engineering hurdles. People are already working on extending these ideas.
So what's next? What's on the near horizon for this kind of work?
Well, there are concrete plans, workshops at major conferences like NeuroIPS, focusing on operator learning for cortical networks. Hackathons plan to try swapping Nobly embeddings with real experimental data. People are starting to test these models on human cortical slice data from epilepsy patients, seeing if they can help predict seizure patterns, and, as I mentioned, integrating them into neuromorphic chip designs.
So it's definitely moving beyond just papers and into practical tools and applications. What's the kind of ultimate vision here?
The vision feels like creating libraries of neurons,
Huh.
almost like, icon packs for designers or component libraries for engineers. You need a certain neuron type. Pull its latent vector, want to tweak its adaptation slightly, adjust the corresponding dimension in its embedding space, and boom, you have a fast, realistic, dynamic model ready to go. It really starts to blur the lines between discovering biological principles in the wet lab and engineering complex neural systems in the dry lab.
A library of plug-and-play biological components defined by their behavior.
Yeah,
That's a really compelling picture.
it is. It holds huge promise, but also makes you think about where that blending of understanding and creation ultimately leads.
Yeah. This whole deep dive into Nobly really hammers home a key insight, I think. Sometimes the best path isn't just more brute force, it's finding the right shortcut, the right tool for the job. By treating that biological messiness, that variability, not as noise, but as signal, and learning it within this continuous interpretable space based on biology, they've really changed the game.
Absolutely. Simulation becomes synthesis, analysis becomes interactive imagination, and suddenly, exploring the cortical zoo is something you can do powerfully on a single GPU.
The field will definitely keep pushing more detail, bigger networks, more complexity. But that core idea, combining biological insight with the right math, like operator theory and modern ML, that seems really powerful.
It opens up completely new ways to both understand the brain and potentially build intelligence.
Definitely. And it leaves us, and you listening, with a fascinating question to ponder. What does it truly mean for understanding brains and for building AI when we can synthesize and explore entire populations of realistic neurons almost effortlessly? Where does that boundary between understanding biology and creating it start to blur next?